Hi guys, we are going to do our notes online today since I can't be with you. We're going to run through these notes as quickly as possible and then give you some more time to work on whatever it is you have for outstanding assignments. Yesterday we dissected hearts and hopefully you got your heart to look something like this picture. The biggest challenge that I find with students, whether it's looking at a picture of a dissected heart or the models that we have from our textbook, is to just figure out which side is which, left versus right. Because depending on how this was cut and whether we're looking at the anterior or posterior section of this, it could be either way. So how do you tell the left side from the right? The easiest way to do this is to draw a line through the interventricular septum and to look at which side is bigger. So remember, the interventricular septum is this wall separating the two large pumping chambers. And if you remember our discussion of the pulmonary versus the systemic circuit, the pulmonary circuit is very, very short and doesn't require as much force or as much pressure. And so the side responsible for pumping to the lungs is going to be much smaller than the side responsible for pumping to the rest of the body in the systemic circuit. So this side of our model is the right side, and this side of our model is the left side. So it's a little bit backwards of what you may think, because this is actually, it has been sliced at a frontal section, and then we're looking at that front or anterior portion of it. And so always look for the thickness of that myocardium, and the left side is always going to be thicker. So today we're going to talk about how the heart actually does its job of pumping and moving blood throughout the body. And remember, we also discussed how all organs have multiple types of tissue. Today we're going to focus on the nervous tissue that is located in the heart and helps it to time and coordinate how the atria and ventricles contract. This timing and coordination is really important because otherwise the blood won't move forward. If they're out of sync or they're working not coordinating with each other, then the blood will just kind of sit there and be stagnant and not actually move forward. So the atria are going to contract first in a cardiac cycle, and both atria, the left and the right, are going to contract simultaneously. This will allow blood to move from the atria to the ventricles through the atrioventricular valves. Then a short time later, the ventricles will contract, and the right ventricle will send blood to the lungs. The left ventricle will send blood out to the body. So we kind of go atria, ventricles, rest. Atria, ventricles, rest. Also remember, we discussed on Wednesday how these valves really help keep blood moving in the forward direction because they're going to prevent the blood from going back up into the atriums, or once it's out into the pulmonary artery or aorta, from it falling back into the ventricles. So all the parts of the heart really do have to coordinate and work together in order for blood flow to be the most efficient. It is possible to live a very long time with a mismatched heart. Sometimes we call this atrial fibrillation, where the atria kind of flutter and don't quite contract correctly. We have ventricular fibrillation, or VFib, and people can live quite a long time with it, but they're definitely not going to have the efficiency that somebody with a properly functioning heart would have. So this is a superior view of the different valves when they are contracting. So the top picture here is when our atria are contracting, the blood is going to flow through these open atrioventricular valves, but then the semilunar valves are going to close. So any blood that was just ejected can't fall back into those ventricles. Then a short time later, we see these big AV valves are going to slam shut. You can see really nicely here why it's called bicuspid, because it kind of has two flaps or two cusps, and the tricuspid has three. So our pulmonary and aortic valves are open while our AV valves are closed. So the way that these contractions are regulated is because the cardiac muscle has really interesting histological traits. So we look at a cellular level. Each cardiac muscle cell is striated, so it has alternating bands of actin and myosin. It has one nucleus, but it has branching fibers. It's almost like it's reaching out and holding hands with all of the adjacent cardiac muscle fibers. This really helps the whole heart work as a unit, and at least the whole chamber work as a unit. It also has lots of mitochondria because your heart never rests. It's always working. From way before birth until the end of your life, your heart is beating constantly. So this is kind of a throwback to Unit 2, actually Unit 2 and Unit 3, when we looked at muscle under the microscope. So we can see these intercalated discs, which are specialized gap junctions that help increase the communication between cells as well, but also hold them really tightly together. And on the right, I really like this artist's depiction of those branching fibers. So some parts of the heart have specialized cardiac muscle fibers that are called autorhythmic. Literally, they cause their own contraction. Skeletal muscles should not do that. They have to have some type of signal, usually the neurotransmitter acetylcholine, in order to contract. But your heart just beats all on its own. We even mentioned that sometimes during heart transplants, as long as the heart is warm and oxygenated, it will beat when it's not even attached to a body, which is really interesting. So the part of the heart that usually sets the tone for how fast hearts are going to beat is called the sinoatrial node, or the SA node. They sometimes call it the pacemaker of the heart. And without input of the autonomic nervous system to either ramp it up or slow it down, the sinoatrial node will tell your heart to beat about 100 beats per minute. So hopefully your parasympathetic system is kind of in charge and your resting heart rate is somewhere in the 60s or 70s. It could even be lower if you're like an athlete and you're in really good cardiovascular shape. Resting heart rates can be in the 50s and it's not an issue. After the SA node, the signal will kind of spread throughout the atria, and then it will cause the atria to contract. From there, it's going to travel to a second node in the interatrial septum called the atrioventricular node. Then it's going to travel through another nerve called the bundle of His, then split into right and left bundle branches, and then finally the Purkinje fibers that penetrate through the ventricular walls. Only once the signal reaches this final step do the ventricles contract. So there is a slight hesitation between the atria contracting and the ventricles contracting, which is actually kind of handy because blood is pretty viscous and it might take some time for it to move. It's not just an instantaneous motion from atria to ventricles. So here's a nice diagram showing all those parts, the SA node, how it causes the atria to contract in unison. Then it also triggers the AV node to send that signal down and around each ventricle. And then the ventricles contract in unison. So we talked about action potentials in Unit 5. So let's just review this very briefly because cardiac muscle works really similarly to other muscle types that we talked about before, and even the action potentials that go through our neurons. So all muscle and nerve fibers have a resting state that is negative, about negative 70 millivolts, and that's due to the action of the sodium-potassium pump. We also know that calcium ions are stored in specialized endoplasmic reticulum called sarcoplasmic reticulum. That'll be important later. Then, as the sodium channels open in response to the sinoatrial node, it's going to cause the membrane potential to become less negative. Eventually, it gets to the point that it triggers a contraction. The sodium channels close, calcium rushes out, and that causes the muscle to actually shorten. Then potassium channels open, the potential is completely reversed because now we have positive ions everywhere. And then eventually the sodium and potassium and calcium will all get put back where they belong by the activity of several active transport pumps. So the heart is always moving, and it's the moving of ions that will actually generate the action potentials that cause heart contraction. So the big differences between the heart and other types of muscles is that the contractions are faster and last longer, which again makes sense. Blood's a little viscous, it's going to take some time to move. So think of it as contracting really fast and then kind of holding it for a second before it relaxes again. We also know that cardiac muscle fibers have a longer refractory period. This is the amount of time it takes to put all of those cations back where they belong, the sodium, potassium, and calcium. This just means that we can't have a second contraction for a certain amount of time. That time's a little bit longer in heart muscles than it is in other types of muscle. So as I mentioned, it's almost like it depolarizes and then it holds that contraction. We call this stage the plateau phase. So it plateaus, or it kind of maintains that contraction, and then it repolarizes. This is just slightly different but it makes sense when we think about the whole point of muscle contraction in the heart is to push blood. So the heart does require a constant supply of energy because all of this is work. The shortening of the muscles is work. So ATP hopefully will be generated in the mitochondria through cellular respiration. That requires a food source, usually glucose, and oxygen. If the heart is ever deprived of oxygen, like it might be during a heart attack, it will trigger a different reaction that creates the byproduct creatine kinase. And so if you've ever heard that somebody's like, oh, they think they had a heart attack, they have to go check. This is usually what they're checking for, is this substance in their blood. This shows that the heart was doing anaerobic or oxygen deprived respiration for at least a certain amount of time. Even the amount of creatine kinase can give us an idea of how severe the heart attack was. So for any heart patient that is looking to see if this conduction system is working, the test they're going to have is called an electrocardiogram, an ECG or EKG. They're the same thing. And what happens is it creates a picture of the nerve impulses as they travel through the heart. It includes 3 recognizable waves, at least if you're a cardiologist, the P wave, the QRS wave, and then the T wave. Let's just look really quickly at this before we run out of time. So this is what a typical heartbeat might look like on a heartbeat monitor. And what this is a tracing of the electrical activity in your heart. The first wave is the P wave. Then we have down is the Q, up is the R, down again is the S, and then we have a little hill that's the T. So if you think of where there's no signal, anything that deviates from this line, either up or down, is considered a wave. And it's handy that it goes in alphabetical order. P, Q, R, S, T. The PQ interval, this time here, is when your atria will contract. And then the RST area is when your ventricles are going to contract. So we can also name these different spots by kind of saying their endpoints. QT is from Q to T. ST is from S to T. And we'll look more at this on Monday when we have a lab all about EKGs. I have some EKG sensors in Wahpeton that we're going to use so that we can kind of practice and see how this works. So here's some other notes about what is happening between each section and what it looks like kind of step by step. Go ahead and pause this if you want to take a closer look. So a whole contraction and relaxation is called a cardiac cycle. And in each cycle, the atria contract, ventricles contract, and then we rest. When the ventricles are contracting, we say that we are in ventricular systole. So this word right here is systole. Say it out loud if you need to a few times. People always want to say systole, but it's systole. During the relaxation, we say that we are in diastole. Okay, and we'll have that in the next slide, I believe. And remember that our ventricles are way stronger and bigger than our atria. So when we talk about blood pressure next week, that systole and diastole will come in handy. We'll also next week use some stethoscopes to listen to our heart sounds. This is called auscultation, just listening. And the sound of the heartbeat actually comes from the turbulence due to heart valves closing. So think of those heart valves kind of snapping shut and creating that little bit of turbulence. We actually only have 2 heart sounds that are typically audible when you're not like a professional doctor, and they're called lub, dup. The lub is going to be the atrioventricular valves closing, and the dup is going to be the semilunar valves closing. So it kind of goes lub, dup, lub, dup, lub, dup. And I'll give you guys some opportunity to listen to that next week. Okay, so at this point, I'm out of time with my recording, but you should spend the rest of the hour working on whatever it is you have to work on. Exercise 20, your term project, chapter 20 homework. Don't forget to turn that in before 11:59 p.m. And I will see you guys on Monday. Hope you have a fantastic weekend.